Thank you very much. It's once again a real pleasure and honor to be here. So Translatum Medicus is a preclinical drug development company, and we focus on macrophage modulation in the treatment of blinding eye disease, first focusing on AMD. We are a J Labs Toronto company. So we all know that AMD is a leading cause of irreversible blindness. Patches of tissue die and relentlessly expand. It's a complex disease. Genetics tell us that complement's involved. Epidemiology tells us that tissue stress is involved. And when you actually look at the tissue, you can see immune deposits as well as immune cells. This tells us it's a disease of the innate immune system. So everyone knows the innate immune system is comprised of complement and effector cells, the macrophages. And at Translatum, we believe that macrophages actively participate in the tissue demise. And you can envision how aggressive macrophages could literally gobble up the tissue. We also believe that in many patients, so when I follow a lot of my own patients, we see that drusen actually regress before atrophy or neovascularization occurs. And we think this might be the local macrophage population, the surveilling population sort of waking up. In Sen Lab's group, they beautifully showed that in tissues with dry AMD, macrophages are found in the tissue, and not only that, one of the key molecules that stimulate and activate and polarize macrophages, MCP1, is actually in the vitreous. And this is remarkable because this is not a disease of leaking blood vessels, that's just a local production. So I want to remind everyone that complement and macrophages are in a constant dialogue. And that catalytic cascade that we talk about is limited to the blood. And it's there to rapidly kill, let's say, invading bacteria, foreign pathogens. But in fact, all the cells in our body are covered by complement. And it's how macrophages can distinguish self from non-self and normal self from abnormal self. And macrophages are basically listening to this, and the way they do it is by having receptors on their surface. They have receptors for complement factor H, 3, C3, C5, and they respond then by changing their behavior. So macrophage behavior is plastic, so you can actually push a macrophage to become very pro-inflammatory, pro-phagocytic. If you push it the other way, it becomes angiogenic, releasing VEGF. And that's all regulated at the transcriptional level with networks of genes that get turned on and off. So our approach, we believe that when macrophages attack to cause GA, it's because they become M1, and our therapeutic approach is therefore to revert them back to neutral. And we actually do this at the gene regulation level. So amongst our molecules, TMI-18 is our lead drug candidate. It actually acts in the nucleus and acts at a master switch for that gene MCP1. So under non-inflammatory conditions, you get low levels of messenger RNA. During inflammation, messenger RNA production goes up, and we inhibit that switch. So I'll just show you what it does in a number of our models. So first, we can show that we dose-dependently reduce our messenger RNA targets as well as the protein. We also downregulate downstream transcription factors, which means we have a profound effect further down, including the inflammasome. We protect tissue, and we protect function. And I think this is remarkable. In a couple of our models of GA-like patch formation and expansion, we can completely block this. And when you think about it, this is also remarkable because we're exposing the tissue to the same stresses, but all we're doing is telling the macrophages to behave, and we can completely block atrophy. And interestingly enough, and it's the big question, is if you push the macrophage too far, would you turn it to angiogenesis? Because we actually work on that master switch, it doesn't happen. And we have not seen any indication in vivo, in vitro, or molecularly of the angiogenic profile. So our team has expanded. I'm very pleased to announce we've added Paul Howes, former president of Bausch & Lomb, and leadership roles in thrombogenics and Innate. Dr. Gary Novak is helping us with our regulatory program, and we've also partnered with an in silico drug design company, Cyclica, and they're helping us with our pipeline. So we're moving to clinical trial. We're planning our first phase 1/2A in dry AMD, and we're following on with other ideas. We've produced our first batch of API, and I'm very pleased to announce we've got 1 year stability on our low molecular weight, and we've confirmed our intravitreal formulation. And so we're preparing for regulatory. So thank you very much. A bit later today, I'll be talking about my other company, how we turn this into precision medicine. Thank you.